Okay, we have a couple of things that we need to get accomplished today. One is to deal with any lingering questions that there might be regarding last week's lab, and then to introduce this week's lab, and then the reading quiz at the end. At the beginning of the lab period this week, you've got to turn in a lab hand-in for last week's lab work. I really want to emphasize to you that it's really important to take those seriously. Don't leave questions blank. Don't leave points on the table with these particular assignments, because they're typically between 10 and 15 points, and there will be enough of them to add up to an equivalent of an exam. And so this is a place where you should be banking some points in case you struggle occasionally on an exam. So take the time to answer those questions carefully. Those are things, too, that you will need to know for the first lab quiz. When I grade these and give them back to you, you should keep these, and then when the first lab quiz is approaching, you should study your old lab hand-ins for preparation for that particular lab quiz. And I'll tell you along the way that there are some things that you will just have to do on the first lab quiz. For example, there will be a question on your lab quiz that will give you some information about, assume you did these particular tests and you got a positive Benedict's and a negative Sudan 3 and a positive iodine test, for example. Well, number one, you're going to have to know what those tests test for and what color changes indicate positive and negative tests. Then you're going to have to identify using a chart, the food chart that you used in lab, what that unknown is. Okay, so you're going to have to be able to do that. That's why it's important for you to do these lab handouts on your own and not just copy down, which is plagiarism, by the way, answers from your lab partner, because number one, that's illegal, and number two, you won't be learning this stuff yourself. Okay? So I'll tell you right now, you're going to have to know how to identify an unknown food given some data and a food chart. Okay? And we'll tell you along the way that there are going to be other things that you're going to have to do as well. So any questions about anything that's kind of lingering out there about that first lab? Anything that's, okay?
Discuss
Question 2 and question 4. Give me question 2.
Basically, says each of the test function groups have control substances and negative test compare. The test for reducing sugars and negative control of water, pseudoprotosis modified and experimental procedure for the lab test. For reducing sugars using a test for the hydroxyl groups instead of a test for the presence of free aldehydes.
Okay. So we detected reducing sugars by doing an aldehyde test. But carbohydrates have hydroxyls too, right? So if somebody could come up with a test for hydroxyl groups, would that be a good test for reducing sugar? I see some people shaking their head no. I hate to just give you the answer because that's one of the things I want you to think about. Okay.
What is this dissolved in? Water. Is there more water or sugar? And put 1 gram of water in 100 grams of sugar. Or 100 grams of, 1 gram of sugar and 100 grams of water. More water or more sugar? Water. Okay, so there's a whole lot more of this than there is of this. Can you do a test for hydroxyl groups and find out how much sugar is there? Why not?
Because water has
Your solvent has all the same things in it.
So anything that dissolved in water, be it sugar, be it protein, would give you a positive hydroxyl test, right?
Yes.
So is water a good control in this case? No, because it gives you a positive test.
If you're testing for hydroxyl.
Yes.
Remember what we said in the beginning of the lab. We're taking advantage of the unique chemical or physical properties of the biological molecule in order to detect it. Hydroxyl groups are not unique to the sugars. You've got hydroxyls in the water. But what isn't unique is the aldehyde. Okay.
Okay. Question number 4 asks about the insolubility of fats, but fats are made up of 2 components, glycerol and fatty acids, both of which contain polar groups. Are substances that are polar generally soluble or insoluble in water? Yeah, there's kind of a dictum in chemistry that says like dissolves like. Is water polar or non-polar? Water's a polar molecule. It's got polar covalent bonds. So typically polar substances are going to dissolve well in water. Well, glycerol has polar groups. Those hydroxyl groups are polar. Fatty acids have carboxyl groups, which are polar. So why is a fat that is composed of 2 polar building blocks not soluble in water?
How do you link this to this? When you make a polymer, what kind of reaction occurs? Dehydration. Where's the water going to come from? You're left with that, an ester group, ester linkage.
So when you pull these functional groups and these hydrogens off, this is going to be unstable because it's not sharing electrons anymore. This is going to be unstable. And things want to be stable. So that carbon and oxygen is going to form a bond. And that's called an ester bond. Okay?
You lose the polarity. Remember? Because you've got a highly electronegative oxygen here, a highly electronegative oxygen here creates a small dipole. Right? When you pull that away, you lose the polarity when you polymerize the molecule. Right? Don't forget there's a long hydrocarbon chain off of each of these fatty acids.
And those hydrocarbon chains are nonpolar.
Correct.
And that contributes to the insolubility of fats in water. That make sense? Another place you don't want to leave points on the table if possible is in the pre-lab quizzes. Did you want to say anything, Larry, about the pre-lab quizzes and how to prepare for them and how to maximize performance on
Well,
them?
after the first one, you should have a better idea of the kinds of things that I expect you to know about the lab. The questions you got were very basic. What are the 4 biological macromolecules? What is this test for? What is that test for? Which is what the entire lab was about. Right? This week, what we're doing is looking at prokaryotic organisms and eukaryotic organisms. I wonder if there's a question there. What's the difference? Right? How do eukaryotic organisms differ? You know, as far as the types that we're looking at. We're looking at autotrophic and heterotrophic. What does that mean? Right? These are all bolded terms in the text. So those are the kinds of things that I expect you to understand. I don't expect you to understand. For example, we're going to add neutral red to the paramecia samples. I don't expect you to understand why. All right? It states in there to do it, and gives you a hint why. That's more detailed than I expect you to know. I need to be sure that you have some concept of what it is you're doing and why. Okay?
So it might be helpful if you did prepare notes, and I would recommend that you prepare notes for these,
Oh, absolutely.
to go back to the notes you prepared now and look at your quiz and say, what wasn't in my notes that was on the quiz? And do kind of a comparison there and see, okay, how can I do a better job of kind of reminding myself what the major kind of elements are in this lab.
And really, there's no excuse for people getting 2s and 3s and 6s when you have a cheat sheet. Come on. the idea of having you write out the notes is because memory is more easily created when you have multiple sensory inputs. So you're looking at it, you're reading it, and you're writing it. That's why we allow you to do the notes. It helps you remember. So take advantage of it. These are freebie points.
Okay, as far as lab is concerned this week, there's really a couple of goals of lab this week. And number one is to have you start looking at some real living cells instead of just pictures and diagrams up here on the PowerPoint slides. And to really see living things in their natural environment. And you can learn a little bit about their behavior. This is also going to allow you to appreciate a little bit the diversity of cells, to look at truly the difference between prokaryotic and eukaryotic organisms, and to understand some basic fundamental differences between different groups of organisms, photosynthetic versus non-photosynthetic, for example, single-celled versus colonial or filamentous forms. And so this is a chance to really do some observation and start applying a few of the things we're learning as we go through the tour of the cell. The other important thing here is to get to be comfortable using the microscope. I know probably everybody in here has used a microscope before, but the microscopes we're going to use in lab are pretty high-end, and while you may have used a microscope for a few things, you may not have used oil immersion, for example, and you might be using the microscope but might not be doing it properly. And so one of the things we really want to ingrain in you is proper use of the microscope. There's kind of a formula that you use to go through and view slides, and if you follow that kind of regimen, you're going to be much more successful than if you don't. If you take advanced courses in biology, particularly if you end up in microbiology as a senior, you will use the microscope all the time, and you've really got to be good at doing it if you're going to see your unknown, for example. So what I want to do now is to give you a brief overview of the microscope and some things you should be looking for, and then Dr. Morden will reinforce some of these things in lab and reiterate some of the parts that are really important. So if you take a look at the side view of our microscopes are binocular microscopes, meaning that there are two oculars. That means that you don't have to squint and hold one eye shut, only looking through a single monocular objective lens. Please take advantage of the fact that these are binocular scopes and use both of your eyes. It's not uncommon to see a student closing one eye and just looking through one eyepiece. That defeats the purpose of these particular instruments. Now, there can be some challenges associated with getting that headpiece aligned properly so that that inner pupillary distance, the distance between the two oculars, is set right for your face. So you can slide those oculars back and forth relative to one another, and doing that so you see a single concentric field of view instead of two fields of view, one with each eye, can be a little bit of a challenge. And sometimes students, it's just not a problem for them. Other times students will struggle with that. So you're going to spend some time with them setting that inner pupillary distance. Okay? Some key elements you can see about the microscope here. Down at the bottom we have a substage illuminator, and that's where the light is going to come from that will shine through this little system called the condenser, and then up through the stage, and then through the through the objective, and then up through the ocular. Okay? This is a compound microscope, and so that means at any given time, the light is passing through two lenses. One of four objective lenses, and you can choose what those lenses are by rotating those lenses around, and then on through the ocular. The four lenses, the four objective lenses, are the 4x, the 10x, the 40x, and the 100x. The ocular is 10x. Okay? So your total magnification that you get when you're looking at something is the product of whatever objective lens you've chosen times the magnification of the ocular. So the lowest magnification we can get is 40. The highest magnification we can get is 1,000. Okay? The 4x is the scanning objective, and that's really used to just kind of find your general place on the slide. And you really can't see much detail by looking through that scanning objective. But it can allow you to position and kind of figure out where you are in the slide. So when you start focusing on a slide, you should always begin on the scanning objective. As soon as you have found something of interest, then you can quickly switch over to the 10x, or low power, up to the 40x, or the high power, and then, if appropriate, onto the 100x, or oil lens. There are some situations with the eukaryotic organisms where it's not really even necessary to use the oil lens. You can see plenty of detail at the 40x or even the 10x objective. If you're looking at prokaryotic cells, then you're going to need that higher magnification that is provided by the oil lens. Keep your ocular lenses clean. They tend to get smudges on them from eyebrows and all kinds of other stuff. There's lens paper in your drawers or out on the tabletop that you can use to clean off your oculars, and also your objective lenses. Those objective lenses tend to get a little bit dirty, and so if your image appears to be blurry at all, one of the things you should do is clean off the lenses with lens paper. This is the on-off switch for the light. This is an illumination adjuster that controls basically the voltage that goes through the substage illuminator. And so by rotating this left and right, you can increase or decrease the amount of light that you need. Different objective lenses require different amounts of incoming light. And if you have too much light coming through, it can literally blind you. Okay? Think about how you can't see when you're driving west in the afternoon and the sun's shining through your windshield. It just makes it almost impossible to see. So if you have too much light coming through this, it can really obscure your image. And so one of the things you need to get in the practice of doing is changing that illumination level, either by this illumination adjustment or on the bottom of this condenser lens, which is below the stage, there is an iris that you can rotate back and forth that works like the iris of your eye. And you can stop down the iris to make less light come through here, or you can open it up to make more light come through. And so that's something you've got to play around with. The stage is a movable stage, and it rotates up and down by use of this coarse adjustment knob. The objectives are fixed. On some microscopes, the stage is fixed and the objectives raise and lower to focus. On our microscopes, the objectives are in a fixed position and the stage lowers and raises to focus. Yep.
A couple of pointers. On the light adjustment, generally you'll be working between number 3 and number 4. If you go above 4, it's way too bright. And this substage condenser, that's okay, this iris down here, usually we're going to be working with it almost completely closed or just a little bit open. If you open it too much, then you won't be able to see, especially the live material.
The substage condenser basically helps to focus the light coming out of the illuminator here. And I don't think you'll really have any occasion to focus that substage condenser. I'll show you where the focus knob is in a second. I think for all practical purposes in lab this week, it is best to have this substage condenser in the full upright position, closest to the stage.
Yeah, all the way up and then take it down.
Okay. Okay, so here's a different view. Here are your two oculars, and then you can, by grasping the base of the ocular down here, you can move them further apart and closer together. And there is a little kind of guide here that can show you, once you've got it set for your eyes, where that setting is. And so if you're using a microscope again, you don't have to go through that process. You can just use these little guides to set the oculars. Over here on the left ocular is something called a diopter ring. And if you've ever used binoculars before, you're familiar with the diopter ring. On binoculars, on one of the two oculars of the binocs, you can change the focus of the usually left side to focus the two eyes independently. Because most of us have differences in our two eyes. Okay, so Dr. Morton will talk a little bit about how you can use this diopter ring to independently focus the left ocular versus the right one to get a good, clear image with both of them. The stage is fitted with a little clip device that you can put your slide into. It's got a little retractable spring here, and if you push this little lever, it pushes the stage clip back. So the slides that nestle into this little angle right here on this metal stage clip, and then this touches the other end of the slide to hold it in place. Okay? You can move around the slide by using these stage movement knobs down here below the stage. One of them rotates it in the X direction, and the other in the Y direction. So you can very precisely move that slide around once it's secured within that set of stage clips. And sometimes you'll see students kind of trying to wedge the slide underneath the edge of the stage clip. It really just springs back, and then the tip of this should nestle against this side of the slide over here, hold it in place. Okay. One of the things that you've got to be careful about with the microscope is the phenomena of optical inversion. Let me know what optical inversion refers to.
Everything you see is flipped.
Yeah. The image that you're seeing coming through the oculars is upside down and backwards compared to the slide. So if you want to see something on the right-hand side of the slide, you've got to move left. If you want to see something toward the top, you've got to move it down. And so that gets a little bit weird to learn how to do until you just get some practice. But remember that phenomena of optical inversion. Down here you can see the coarse focus and the fine focus. And these are the knobs that move the stage, large amounts or small amounts. It's hard to see the iris right here, but the iris again can be, it's a little ring that can be rotated to let more or less light in. Here is the formula you should always use for looking at slides. Let's say that we're using a prepared slide. A prepared slide has a cover slip permanently fixed to it. Okay? When you start, you should always be on the scanning objective. And when you put your microscope away, it should always have the scanning objective in viewing position. The stage should always be fully down. Okay? Put your slide into the stage clip, and there's a little hole in the stage that the light shines up through. If you center that cover slip, which is typically round, might be square, right over that hole, then the light's going to be passing up through it, and you're going to be looking at what you want to look at. Okay? Then, before you look through the oculars, use the coarse adjustment to raise the stage to the full upright position. As close to this objective as it'll go. Now, it's set to stop before it runs into the objective. Then, and only then, can you start looking through the oculars, and then you focus downward by moving the stage away from the objective. You should always focus downward as opposed to starting with the stage here and moving it toward the objective. Why? Because you don't want to run the stage into the objective. So have the stage in the full upright position and then focus downward using the coarse adjustment. Once it comes into fairly good detail, you can use the fine focus then, which moves the stage very small increments. Okay. Once you're in focus on the scanning objective, then slide it over to the low power. At that point in time, you should not have to touch the coarse focus to get into critical focus. You should only have to use the fine focus. Once you've seen what you want to see on low power, rotate the lens over to the high objective, 40x. Again, you shouldn't have to use the coarse adjustment. You should just have to use the fine focus. And then I'm going to let you tell them about oil. Okay? So if you need to use oil for the prokaryotic cells, then there are a couple of additional things you need to do. But starting with that process of starting on scanning, focusing downward, low power, refocusing with the fine focus, high power, is something that you should just really get in the habit of doing all the time. What else have I forgotten to tell them?
Okay. A classic mistake that students make is they'll get something in focus with the 4x objective, switch to the 10x objective, and then things aren't really in focus too much, so they start moving this fine focus and they start throwing it all over the place. Well, that means that your focus with the 4x probably wasn't very good, because once you switch from one lens to the next, you shouldn't have to turn that fine focus more than one revolution in either direction to get the sample in focus. If you have to do more than that, then go back and start over again, because otherwise there's a limit to the travel, how many times you can turn this fine focus, and if you push it too much to one edge or the other, you're going to lose the range to work with. When you go from 40x, from the 4x to the 10x, you can pretty much leave your light setting the same. You won't see too much of a loss of light. But when you go from the 10x to the 40x, from the low power to the high power, usually you have to open your focus diaphragm a little bit and maybe increase the light intensity a little bit. Right? You do not want the background to be bright white. You want it to be sort of cream colored, because otherwise you're putting too much strain on your eye. And one of the things we'll start off with this afternoon is how to appropriately set not only the interocular distance, but the difference in focus between your eyes. Because if your microscope is appropriately aligned to your eyes and focused appropriately, you can work on the scope for hours and you won't get headaches. Most kids talk about, I can't use a microscope, it gives me headaches. Well, it's because they haven't set it up properly. So we'll spend some time in the beginning of class taking you through the process of setting it up properly. You will all have your own microscope. Okay, so there won't be, sharing, looking at images. Once you've set up your microscope, and be sure to set it up right in front of you, right in the middle, and fairly close to the edge of the desk, do not move it. A lot of times people will call me over and say, can you look at this? And then they scoot it across the bench top. Well, that just throws everything out of focus because it's just bouncing. So don't do that.
Another thing that you can do and should do is your lab stools will raise and lower.
Yes.
And it's amazing some of the contortions you see students do to try to get to the point where they're a comfortable viewing height without actually raising and lowering the chair. So they've got their legs under them and they're,
Those of you who are vertically challenged, the chair actually does go up quite high. For the tall ones, there's a limit to how far it'll go down. I can't help you there. You just have to stoop over a little bit.
There are some challenges that Dr. Morton will talk to you when you start viewing wet mounts, which are living materials, and making your own slides, but I'm going to let him deal with that during class.
Yes.
Okay. We're not using Hydra today, right?
Yeah, unfortunately.
Our Hydra did not come, so I guess I don't need to say anything about the stereoscope then. That's too bad. Okay, so some of the things you're going to be looking at are some oral bacteria, some teeth scrapings. Here's a slide of something typically that you might see. This is a nice colorized picture of what microorganisms look like, but each one of these individual little dots right here represent individual bacterial cells. And so you'll be a little bit grossed out about the bacteria that you have living in your mouth.
Yeah. I would suggest after lunch that you not brush your teeth because we really want the stuff that's around the edge of your teeth and on the cells on the inside of your cheek. So, give your mouth a break for one period.
Oops. This is an interesting organism you're going to look at. Most of you have probably seen amoeba before. Amoeba is able to do something quite interesting called amoeboid movement, where the cytoplasm starts flowing and it basically kind of drags itself along. This is the same kind of movement the white blood cells use to patrol around your body and look for foreign invading things like bacteria. So you can see some cellular detail with this amoeba. You can see a nucleus here before. You can see some vacuoles within the cytoplasm. Amoeba is classically an example of less is more in terms of light. And one of the things you really have to do to see amoeba is to almost close your iris completely, because the contrast between these unstained cells and the background is really hard to see unless there's just a small amount of light coming through.
When you do this, you won't see this color, but you will see dark, almost black vesicles streaming around, usually circularly in the cytoplasm, especially out through some of these pseudopods.
This is a prokaryotic filamentous microorganism. This is obviously photosynthetic. You can see the bluish green pigment in these particular cells. You'll notice here too that there's a slight amount of specialization. All these cells are pretty much identical, but occasionally you will see this kind of balloon-like cell that's part of the filament. That's a kind of unusual in prokaryotes, that there's that level of specialization occurring within this filamentous organism. That's Anamina. Nitrogen fixation. Most of you have probably seen Paramecium raccool. You can barely see along the edge right here the little cilia that make this a modal organism. Because this is modal, it's a challenge to keep up with it and to be able to follow it as it moves around. And so there is some sticky stuff called protoslow or detain that we're going to use to kind of slow these organisms down. But again, you can see some intracellular detail right here.
Okay. These move too fast for you to be able to follow them with, on the microscope. They'll just zoom right across your field of view. So as Dr. Browder mentioned, when we prepare the sample, we will add this stuff called detain. It's derived from cellulose, and essentially what it does is make the fluid around the cell really thick so that it can't move quite so fast. The other thing we're going to do is we're going to add a drop of red dye, which the paramecia will take up very quickly and store in vesicles. All right, so that'll help you see where the cells are, as well as you'll be able to see things moving around in the cytoplasm.
And I believe you're gonna have some of these images available, aren't you,
Yes.
during lab? Because sometimes it's just hard to know what you're looking for unless you have an idea of what it is.
It's a footprint.
Yeah.
That's what it looks like. These are huge ones too.
Yeah. Here's a small colonial grouping of 2 to 4 cells called Protococcus. Can't see a lot of detail in them, but they are obviously photosynthetic organisms. I suppose if you look really hard, you might be able to kind of see the chloroplasts a little bit, but that's about it. Another colonial microorganism where it's easier to see the chloroplasts in these cells. They typically are little packets of 4 with these interesting little spines sticking off of the end. Cenodesmus. Volvox are a very beautiful organism. They're also a colonial organism. And each one of these little dots right here represents a Volvox cell that are massed together in this kind of gelatinous ball. And then these are little daughter colonies of Volvox that are forming within the mother colony right here. And sometimes you'll see the mother colony broken open and some of those daughter colonies being released. These outer cells in the ball are flagellated, and so it's not unusual to see this ball rotating around as those flagella beat back and forth again. I'm not going to have Hydra. It's too bad. Hydra are interesting multicellular organisms. And then if we can find them, there's going to be some intestinal tissue that we're going to look at. This is a section of intestine, and this is the cavity or the lumen of the intestine. And when you start looking at this in more detail, you can see several cell layers There's a muscular layer down here. And then there are these little projections called villi that wrap back and forth to increase the surface area for absorption. And then if you start looking at the individual cells within these villi, you can see that these cells right here sometimes are different from one another. Some of these cells, called goblet cells, secrete mucus into the lumen of the intestine. And then some of those cells are also have a very folded cell membrane, which again increases the surface area on those villi. So on this slide you can see some of the muscular layers, the villi right here. So we're going to have you look a little bit about some of the specialization that occurs in some of the tissues of multicellular organisms.
One of the overriding themes today is increase in complexity. We start out looking at prokaryotic cells that exist individually or in chains. Then we go to an individual eukaryotic cell like the amoeba. Then we go to photosynthetic cells that live in small colonies or larger colonies. Right? And then you see something like the Volvox, where you have large colonies of cells where some of the cells have different functions to make those daughter colonies inside. And then sort of the most highly developed system where cells have different functions is in this vertebrate intestine.